you. Is this working? Yeah, good. Thank you very much for a nice introduction, and thank you also for inviting me here to speak. Also, thank you for the previous speaker for providing a good introduction. Although it was in Slovak, I was able to follow via simultaneous translation. So I know it has been a great and excellent introduction of the story that I would like to tell you. As said, my name is Anneke Reinierse, and I'm a scientific program manager for the Nutrition Research Center. And with this presentation, I would like to give you an overview of all the clinical but also preclinical data that we have gathered for the evidence for the reduction of allergy development in high-risk infants. I would like to start off by highlighting the importance of the gastrointestinal tract, because beyond, of course, the obvious digestive function of the gastrointestinal tract, it is also known that the gastrointestinal tract is the most immune organ, especially also in early life. And it's not so surprising if you think actually about the surface area of the gastrointestinal tract having 32 square meters exposure to the outside world, because everything that we eat and that we swallow is the outside world. So the exposure to the outside world in the gastrointestinal tract is actually the largest of our whole body. So exposed to the outside world is actually tucked away inside of us. So therefore, it's not so surprising that it's 70 to 80% of our immune system resides in the gastrointestinal tract, because the immune system is trained and programmed to distinguish harmful from harmless and to also defend the body from the outside world. There are several different immune organs present in the gastrointestinal tract, and especially early in life, there is where the high training of the immune system takes place. So it's very important to have an optimal development early in life. But besides having the vast majority of immune cells and immune system resident in the gastrointestinal tract, there's also a very big community of gut microbiota present. And this is as large as around 100 trillion bacterial cells, which is a number that we cannot even comprehend, but it outnumbers the human body cells, and it takes up around 5% of our body weight. So it's a very big community that we carry around every single day and that we also need to take care of and also need to take into account when we think about creating the most optimal development and health later in life. And with this picture, I would like to further highlight how this is all interconnected, because we know that from birth onwards, the immune system, but also the microbial community and even the gastrointestinal physiology is still very immature, and it needs to further develop while also the child is aging. And it takes up to 2 to 3 years of age to reach its full potential. And this is what we often see also as a window of opportunity to help and stimulate this optimal development of both the immune system and the microbial system to be able to give the best start in life and to further promote health development later in life as well. And as said, there's a lot of evidence already known on how the microbial community is interacting with the immune system, but also more and more evidence is coming that the immune system and the mucosal environment is also having an effect on the microbial composition in the gastrointestinal tract. So what do we actually know about how we can influence the microbial composition early in life? We know actually that human milk heavily influences the development of the gut microbiota already from the first instance and the first exposure to human milk. And if we look at the golden standard for reference being a healthy vaginally born infant, when this infant is exposed to human milk, you see that the microbial composition comprises predominantly out of bifidobacterial species. There are also other species, but it's the bifidobacterial species that resides in the infant gut and is also very well capable of colonizing and retaining in the infant gut. But there's more to human milk than only the activity, because there's a lot of components, of course, in human milk that stimulate optimal growth. And the main building blocks of human milk is, of course, water, but the other components are fat, lactose, and proteins that stimulate growth and immunity and gut health. But also a very important building block are the oligosaccharides. And these are known as to be prebiotics and stimulating the healthy growth of the beneficial bacteria in the gastrointestinal tract. Then, of course, there are also a lot of micronutrients like hormones, vitamins, even living cells and nucleotides and minerals that are very important components of human milk that further stimulate optimal development of the immune system, microbial system, but also the metabolic system. And very recent scientific research has demonstrated that even live bacteria are present in human milk. They are there in small amounts, but they're definitely there, and they also come with the dedicated metabolites. And it's only since very recent that it has been discovered that human milk contains about 10 to the 3rd to 10 to the 6th bacterial cells per ml. And they predominantly are dominated by species like the Staphylococcus, Streptococcus, Propionibacterium, and Bifidobacterium. And it's anticipated that these bacterial strains are important to defend against infections, but also contribute to the maturation of the immune system. And this is where we also, as nutrition research, take our inspiration from, because we still see human milk as the golden standard to support optimal immune and microbiota development. And this is why we have obtained a dual approach of active allergy management. And it's already been kindly introduced by the previous speaker, that nowadays there's a paradigm shift from allergen avoidance towards tolerance development and allergen exposure, but also by means of setting the right environmental conditions of the mucosal system to also further support this tolerance development. So therefore we're taking also a dual approach here by controlling the exposure of the protein by means of hydrolyzing the protein and thereby reducing the allergenicity of the protein, however, maintaining the capacity for this protein to be inducing tolerance. And then next to that, we aim to set the right environmental conditions for the gastrointestinal mucosa to support this optimal tolerance development. And today, I would like to take the opportunity to guide you through the evidence that we have obtained throughout the years to support this concept of the dual approach. And I would like to start off with providing the evidence on the prebiotics and the probiotics. We call this concept, the combination of the prebiotics and the probiotics, we call this the symbiotic concept because it provides the best of both worlds. If you consider the prebiotic as a favorable substrate for a probiotic bacteria, meaning that the probiotic grows very rapidly and fastly on these prebiotics, and the probiotics are beneficial bacteria that bring a health benefit to the host, it's easy to see that a combination of those where they are stimulated is a symbiotic concept. In our formulation, we have a prebiotic mixture of short-chain GOS and long-chain FOS, and this mixture, in a specific ratio of 9 to 1, was selected to mimic also the composition of human milk. And then next to that, we selected the Bifidobacterium breve M16V as the probiotic strain to go into our formulations. So why did we select Bifidobacteria M16V? First of all, why did we select the species of Bifidobacterium breve? We selected this species because it's the most commonly isolated bifidobacterial species from human milk. We know that Bifidobacterium breve species are present in human milk. And then next to that, we also know that this species is naturally occurring in the infant gut. So once this species is colonized in the infant gut, it will remain in the infant gut and it will proliferate and grow. So this is the reason why we selected the Bifidobacterium breve species. And now specifically on the M16V strain. We selected this strain, first of all, because not only by us, but also by the supplier where we buy this strain from, it was demonstrated that this specific strain is safe in the use for infants. And this was definitely demonstrated also in the extremely fragile infants, like being the premature infants. Then next to that, we have selected this strain for its potency to have immune modulatory effects in both clinical and preclinical settings. And then last but certainly not least, we have demonstrated that there's clinical efficacy for this specific strain to reduce the allergic symptoms in infants. Hereby, I would like to show you some data on the lab animal trials that we have performed with the symbiotic mixture. You can see in the left graph that we have tested both the prebiotics, the probiotics individually, but also the concept together, the symbiotic concept, in a cow's milk allergy model. And it's very clear from the picture, so in the green most right bar, you can see that the symbiotic mixture was best capable of reducing the cow's milk allergic symptoms in these animals. Since we're also very interested to see what exactly is the mechanism behind, so how does the symbiotic come to this effect, we have also demonstrated that the symbiotic supplementation is associated with an increase in galectin-9. And that's what you can see in the second graph. You can see that the symbiotic mixture induces a significant increase in galectin-9. And it's known that galectin-9 is a sugar component that can specifically bind IgE and thereby block the allergic reaction. So this could be a potential mechanism on how the symbiotics can reduce the allergic effects. And there's more research programs going on in our center to further dig into the mechanism behind the symbiotic effects. We have also performed several clinical trials with the symbiotic mixture. And here I would like to highlight our trial in elective C-section delivered infants. As was also mentioned by the previous speaker, we know that C-section delivery goes with a disturbance and a disbalance in microbial composition, which is often also referred to as dysbiosis. And in this trial, we aimed to re-establish and restore the colonization and the dysbiosis in these infants. And as you can see from the graph, you can see that infants born via C-section and exposed to symbiotic mixture, we were able to restore the microbial composition in these infants by means of measures of bifidobacteria. And that we could restore this to the level of what we said as our control group being the vaginally born infants. What you can see is that also there was a big, let's see whether this worked, yeah. There was a big gap between the C-section delivered infants, the vaginally born infants and the C-section delivered infants. So you can definitely see that C-section delivered infants have a lack of bifidobacteria, which we were able to restore by supplementing them with a symbiotic mixture. Then next to that, we also demonstrated that there was a favorable milieu for the defense against pathogens because we demonstrated increased levels of acetate and a decreased level of fecal pH, which is known to reduce the risk of infections. And then last but certainly not least is that we saw a potential protective effect on skin allergic symptoms and especially eczema. And it's something that we didn't really look for in this trial. It's also something that we did not really design in this trial, but it's something that popped up in the adverse events. So in the infants that were exposed to the symbiotic mixture, we saw in the adverse events that there was less allergic skin symptoms and less eczema. So this is definitely something that we would like to follow up on and have a dedicated clinical trial to look into the effects of C-section delivered infants. Then to come to the second part of the dual approach of active allergy management, I would like to highlight a bit more on the concept of controlled exposure. Oh, yeah, thank you very much. By means of the hydrolyzed protein and reducing the allergenicity while maintaining the tolerogenic capacity. And therefore, I would like to take you a little bit back into the past. It has already been said by the previous speaker, but in the past, life was actually pretty simple. It was all about avoiding the allergen and thereby preventing sensitization. So by means of hydrolyzing the protein, there would be no response, no provocation of the immune system, and therefore no allergic sensitization. And it was this line of thinking that actually came to the design of several different trials, ended up in meta-analysis, Cochrane reports, and a lot of position papers and guidelines. And if you pile this all together, it was 19,000 infants that were studied, but today there's still no consensus in the guidelines with respect to how should we recommend and position the partially hydrolyzed protein. And this is because actually life was not as simple as it was initially thought. Because if I take you to this very simplified timeline, in 2006 it was the old line of thinking, avoiding the allergen, avoiding a reaction of the immune system, and thereby avoiding sensitization, and just wait and watch for the development of tolerance simply by just crossing the fingers, so to say. So, but what we know now is actually that true allergy prevention is actually the acquisition of oral tolerance. And oral tolerance is the default setting of the immune system, and it's a conscious training of the immune system to tolerate harmless components. But to be able to train the immune system, you need exposure. And this line of thinking is also what has introduced several different concepts a few years ago, where there were several different attempts and activities to have an early introduction of these allergens. So, and I think most of you are familiar with the big trials like the EAT and the LEAP trial, where they at a very early age, even from 3 months onwards already, have introduced infants to several different very harmful or very potentially harmful food allergens. And it's also the increasing knowledge of the role of the microbiota that also came into play, understanding also that it's more and more complex to study allergy prevention. And next to that, it's also known that it's not only exposure to the protein that is important in stimulating the immune system and an optimal microbiota composition, but it's also, there's also a role to play for the additional ingredients, like the prebiotics, the probiotics, but it's also, you have also the different LC-PUFAs that are known to have an effect. So where are we now today? So where we are today is that actually some of these early active introduction is making it into guidelines, because especially the results from the LEAP trial has led to new recommendations and guidelines for the introduction of peanut. There's also a wider acceptance of baked egg and baked milk for infants that have already an established food allergy against hen's egg or against cow's milk. And it's also known nowadays that hydrolyzed formulas actually should have specific capacities towards allergy prevention. So not every hydrolyzed formula is the same. It's the balancing act by reducing allergenicity, but still stimulating this oral tolerance response. So what exactly does it look like? I have mentioned this already. It's like the true allergy prevention is the acquisition of tolerance, but what does it look like if you break it down from a mechanistic perspective? Because it's a different protein recognition pathway. And this is a very simplified scheme, but if you take a food protein, and in this case I took the example of milk, and you expose this to the immune system in the gastrointestinal tract, there are antigen-presenting cells, and in this case I pictured the dendritic cells that can take up this antigen, digest it, and then present it in fragments on the membrane by specific receptors. They are then recognized by naive T cells. And in case this recognition is seen as a harmless recognition, this naive T cell will continue to develop into a regulatory T cell. And this regulatory T cell is very important in stimulating the favorable response towards tolerance induction. This is what is happening in infants, in subjects that are not allergic. This is the default setting of the immune system. But sometimes, and that can be coordinated by the mucosal environment, the naive T cell recognizes this fragment from a food protein as being harmful. And this is when the naive T cell develops into a Th2 cell, which will stimulate the B cell to produce antibodies, and then you get an allergic reaction. So this is what we call the atopic allergic recognition pathway. It's a different pathway. So what can hydrolysis of an intact protein and thereby controlling the exposure do about steering the recognition pathway in the right direction? So if you take a milk protein and you cut this by means of enzymatic hydrolysis, you end up in a pool of several different fragments of this protein, which we call peptides. And here in this picture, in the pinkish color, you can recognize the allergenic epitopes of this protein and later on the peptides, and the tolerogenic epitopes. And ideally, by means of hydrolysis, you cut the allergenic epitopes, but you maintain the tolerogenic ones. So you maintain the capacity for the body, for the immune system, to recognize the fragments from the protein to be derived from cow's milk, so that the immune system can activate a tolerance response against cow's milk. So thereby, ideally, from an hydrolyzed protein, you reduce the allergenic potential, but you maintain the tolerogenic potential by keeping these tolerogenic epitopes. And this is also an approach that we looked for in our research center. And this is a bit of a theoretical approach when we started off our program on tolerance-inducing sequences. What we did is that we took the structure, amino acid sequence from beta-lactoglobulin as being one of the main components of whey protein, and we cut it down into several different peptides of 18 amino acid length with a 12 amino acid overlap. So we cut the full protein down into peptides. And then we started off to investigate mixtures of these peptides, which you can distinguish here in the bottom. Mixtures of these specific peptides. We investigated them for their capacity to induce T-cell proliferation. And by this means, we could see whether these specific peptides were actually capable of inducing an immune response. And it's the mixture, these two mixtures of 1 to 3 peptides and 4 to 6 peptides that demonstrated that they could actually induce an immune response. However, this assay does not tell you what kind of response. So in the end, we selected 3 specific peptides, and we went to an animal model for cow's milk allergy. And we tested them for their capacity to reduce allergic symptoms. And you can see from this graph that this specific mixture of 3 peptides could reduce the allergic response. And when we added prebiotics to it, you can see that it even was capable of reducing even further. So this demonstrates also that setting the right environmental conditions is also stimulating the favorable immune response. Then, of course, we were interested to see whether these specific sequences that we identified via theoretical approach were also present in a partially hydrolyzed formula. By means of mass spectrometry, we were able to identify the specific tolerogenic peptides in our formula. In this top box, you can see the sequence, part of the sequence of the beta-lactoglobulin protein. And in blue, you can acknowledge the sequences that were responsible for the tolerogenic capacity. And over here, we have been able to identify 6 specific peptides that are present in our partially hydrolyzed protein formula that are overlapping with the specific sequence that has this tolerogenic potential. So this means that the partially hydrolyzed protein formula contains peptides that have sequences with tolerogenic capacities. But it's not only that we did, because next to identifying the peptides in our formulation, we were also very interested to see whether they were actually also presented to the immune system. So what we did is that we took this specific partially hydrolyzed protein into an assay where we exposed the protein fragments to an antigen-presenting cell. This antigen-presenting cell then digested and presented it on the membrane, where we isolated it back again, and then we measured it again via mass spectrometry. And in this case, we could identify the exact same sequences that we were able to identify in our formulation to be also presented to the immune system. So it means that the immune system can also recognize these fragments. And then last, we also looked at whether there was a biological activity around this. So we exposed these specific fragments from the partially hydrolyzed protein, again, to an antigen-presenting cell in co-culture with a T cell, and we saw that the T cell started to proliferate. So there was also a biological activity of these specific fragments. So this was the very preclinical work that we did, but we also have performed a clinical trial. And as the last part of my presentation, I would like to guide you through the data that we've obtained in this clinical trial, which is called the PATCH trial. The PATCH trial is a trial to study the prevention of allergy through cow's milk hydrolysate, and the objective was to have a dietary intervention with the partially hydrolyzed formula. I'm losing my screen. There it is. With specific prebiotics versus an intact cow's milk formula. It was a very big trial. We had over 1,000 infants at risk of allergy by means of family history of allergy. We performed this trial in 3 geographical locations. We were in UK and Ireland, Singapore, and in Australia. And we had 10 recruiting centers. We allowed a mixed feeding regime throughout the study. And the primary objective was the cumulative incidence of eczema at 12 months in a predefined subgroup of infants that started the formula already at 4 weeks of age. We also had exploratory parameters by means of exploring the immune mechanism behind and the microbial composition at 6 months. And we also did some post-hoc analysis on the cumulative incidence of eczema all the way up to 5 years in a specific subgroup. With this slide, I would like to acknowledge already the people that have been involved in this trial. So this trial was headed by the principal investigator being John Warner in collaboration with Robert Boyle. In Ireland, the main investigator was Jonathan Hurricane, and in Singapore it was a collaboration between Wen Ching Chang and Mai Wing Chua. And in Australia, the lead investigator was Mimi Tang with quite a big group of investigators behind that. And there was also quite a big group of people working in the Nutrition Research Center. So with this slide, I would like to show you how big this trial was. I guide you here through the numbers of the randomized subjects. So we had 370 subjects in the control formula group, and we had about 350 subjects in the test group. And then if you look at the predefined group where we had the primary outcome analysis, we had 324 in the control group versus 293 in the test group of hydrolyzed protein and prebiotics. So quite substantial amount of subjects. But then when you looked at the primary outcome, and I'll show you here the survival rate of our outcome with the age in days on the x-axis and the percentage of infants on the y-axis, it's very clear from this picture that there was no difference between the control group and the active group. It's needless to say that we're a bit disappointed to see that there was nothing happening between those 2 test groups. However, I'm on purpose also showing you the survival curve because you can clearly see that something is happening in this period. And this was what was drawing our attention because it coincides with the timing of introduction of complementary feeding, which is at 4 months equaling 18 weeks of age. And this is also what triggered us to go back into the database and look whether something was happening there. And we found out that actually only 45% of the infants were following the recommendation of introducing complementary feeding after 4 months of age. So that means that 55% of the infants started to introduce before. And this is something that we did not take into account when we started off creating the protocol because we actually assumed that people would adhere to international and local guidelines. But it was 55% of the infants that didn't. So knowing also at the time that there were several different confounding factors that could influence allergy prevention knowing that also the environmental factors and the influence on the microbiota has a huge impact, we decided to only look at the subgroup of infants that introduced according to the recommendation. Here I show you the numbers again, knowing of course that this was a big trial. We ended up, if we only looked in this subgroup, with still quite a substantial amount of infants that we could study. So it was around 160 infants for the 12 months and 18 months, and almost up to 70 for the 5 years of age. And then when we looked into the survival curve, we saw that there was a significant difference between the test and the control group. So there was less incidence of eczema in the infants consuming the partially hydrolyzed protein with prebiotics versus the intact protein. So these data do demonstrate that it's very important to take into account confounding factors that can influence allergy development. And in this case, this was the timing of introduction of complementary feeding that we know now has also a very big impact on microbial composition. Then we also looked at the mechanistic effect, and we saw that there actually was an increase in regulatory immune cells at 6 months after the intervention. And it was a significant increase in dendritic cells and in regulatory T cells. And if you remember what I showed you on the mechanistic aspect of the allergen recognition part, these cells are very important in steering towards the right recognition pathway of tolerance development. Then secondly, at 6 months, we also looked into cow's milk IgG1 levels, and we saw that there was a huge reduction of cow's milk IgG1 in infants that consumed partially hydrolyzed protein with prebiotics, which might not be so surprising because IgG is often considered as being a marker for exposure to protein and not considered desensitization marker like IgE is, but it's known that IgE development precedes the development of IgE and is associated with allergy development later in life. What we also saw is that after 3 years, this reduction of cow's milk IgG1 was still present. So the exposure to hydrolyzed protein with prebiotics very early in life did have a beneficial effect, long-term beneficial effect on the development of IgG later in life as well. So to conclude on what this trial has demonstrated us, but overall what the data have shown us, is that nutrition definitely can still be an effective way in preventing allergies early in life. But that we should really look at it from a different angle, because it's very important to have exposure to the protein, but then also to specific protein, specific fragments of protein with specific capacities. And we should not dismiss the interaction with the microbial community and take into account the confounding factors like timing of introduction of complementary feeding, but also other factors like a C-section delivery or antibiotic use. So allergy prevention, studying allergy prevention is not as simple as we have been thinking in the past. I've also demonstrated to you that not all hydrolyzed formulas are the same. And like I already said before, it's very important to look for the specific capacities, that not only the reduced allergenicity should be looked at, but definitely also the capacity of the hydrolyzed formula to stimulate and support tolerance development. And where breastfeeding is not possible, the unique partially hydrolyzed whey formula with prebiotics can be an effective approach to reduce the risk of eczema in this specific subgroup of infants that did not introduce complementary feeding. So again, another trial to add up to the already 19,000 studied infants. And still, there's a lot of questions, and still I'm saying that we're in the need for well-designed studies to further integrate in the understanding of today. And it's therefore that I also would like to introduce our clinical trial program for allergy prevention, because we have a big ambition to study this partially hydrolyzed protein with the symbiotic mixture for its capacity in reducing the risk of allergy in infants at high risk of allergy. And we have started off studying this specific hypoallergenic concept in 3 different trials. There's one growth trial currently ongoing in China. That's the Dragon trial. It's the first time that we're doing a clinical trial in China, so we're learning as we go. But it's going really well, and we're aiming to have the last subject in this year. And then we have two very big trials, which is the Tempo and the Maestro trial, which are looking into the efficacy of the partially hydrolyzed protein with symbiotics. The Tempo trial is predominantly looking into the microbial composition or the influence of microbial composition, whereas the Maestro trial will look into the effect on the clinical manifestations. And these two trials are in design exactly the same. So it means that by this we can create very big cohorts for this specific mixture to be investigated. And the Tempo trial, I know there are several different people probably in the room that are involved also in this Tempo trial, because we're conducting this trial also here in Slovakia. And I have to say you're top recruiters for this trial, so thank you very much for that. And it's with great pride that I can also say that we have actually included the last subject for this trial last week. So thereby we can close the number of 850 subjects into this trial. And with all this, I hope that I have been able to give you a good overview on the approach that we have, the dual approach of active allergy management by means of the hydrolyzed protein and the symbiotic mixture. And I would like to thank you for your attention.